Hi, everyone. It's Dr. B here to talk to you today about quantum biology from photons to physiology. This talk is a summary of some talks that I gave earlier this month, first at the Gordon Research Conference on Quantum Biology in Tuscany, Italy, and a second one at the big American Physical Society's Global Physics Summit in Anaheim, California. So without further ado, I'm excited to talk a bit about why I feel quantum biology is an uncharted frontier, not just for biology, but for physics. Albert Einstein, in a letter to Leo Szilard, said that one can best feel in dealing with living things how primitive physics still is. Now, I believe that that is still the case, and I'm interested to explain how these horizons of quantum biology also present new opportunities for physics research. I believe that in addition to developing new insights into the fundamental principles of life and an enhanced understanding of essential biological processes, that in our study of quantum biology, we may learn about physical effects not predicted by standard quantum theory, and we may even encounter challenges to the existing quantum classical paradigm. Now, this isn't just my idea. Certainly, we have the quote from Einstein in the previous slide, but also Erwin Schrödinger and Niels Bohr felt that there might be an opportunity to discover new laws of physics in the study of biology. Now, why would this be the case? First of all, biological systems are open systems, and the canonical quantum theory is framed using a closed, so-called Hermitian mathematical framework. So biological studies are necessarily studies of open, non-Hermitian systems. And of course, all systems are ultimately open to the universe. We can't put a lab in a jar. Can't put the universe in a jar. And so even though we often study quantum physics in closed laboratory environments, in fact, we need to remember that in real life, everything is ultimately open to the universe. Now, the theory of open quantum systems is usually posed in terms of Markovian master equations, where there's weak coupling between the system of interest and some so-called Markovian bath or reservoir. This means that the bath has no memory in itself, that it is constantly returning to some equilibrium state, although we can generalize this kind of picture that's usually presented in terms of the Redfield or Lindblad equation, that is the completely positive trace-preserving map, in terms of a more general equation known as the Nakajima-Zwanzig equation, where there's still weak coupling to a thermal reservoir, but in this case the bath has a memory that can be accounted for in terms of a memory kernel in our simulations, our mathematical model. Now, when we look at living systems, they're certainly open systems, and they may or may not be weakly coupled to a surrounding environment. Now, living systems are, however, generally, I would say universally, warm, wet, and noisy, and this is in part due to the quantum effect of Brownian motion. Now, this is an exciting topic because it was, in fact, the topic of the second of Einstein's famous papers of 1905. Now, it was very important because at that time, the atomic theory, that is the quantum theory of matter, was not yet fully accepted. And subsequently, Jean-Baptiste Perrin won the 1926 Nobel Prize for experimentally verifying Einstein's theory to demonstrate the existence of atoms. Now, this was one of the last straws that convinced many of the holdouts against the atomic theory of its reality, and it was very important in the ultimate development of the quantum theory of matter. Now, quantum effects in water have come a long way, or at least the study of them, since Einstein's time, and the electronic structure of liquid water is in fact highly complex. Far from washing out quantum mechanical correlations with all the noise, in fact, calculations have only recently been able to predict certain properties of water. You might have studied in chemistry class the ionization potential and the electron affinity of water. Now, these calculations have only recently been done in the last 1 or 2 years, to be honest. And so one should not think that water is in any way a trivial system. In fact, the quantum effects in water have taken 100 years in order to fully characterize, and studies still continue to this day. Now, quantum coherence is an interesting topic in quantum biology, and it's certainly been important for me to develop a better understanding of what we really mean when we talk about coherence, which is a definite phase relationship between, well, typically oscillators. Now, coherence can be static. Quantum coherence can take the form of entanglement, and it can also be dynamic in the form of oscillations. Electron tunneling during long-range electron transfer is found to be synchronized for efficient energy conversion. I'll talk a bit more about that later on. And we will find that both decoherent or so-called incoherent tunneling, as well as coherent resonant electron tunneling effects are relevant for biology. So biological systems really span a wide range of different types of coherence, both classical and quantum. Now, if we look at quantum effects in a molecule like ethane, even though it's just a very small, simple molecule with 2 carbons and 6 hydrogens, simply studying something like the torsional energy of the rotation of the molecule with respect to its main axis, as shown here, this kind of simulation can take a whole room full of computing power, like shown in the Sunway supercomputer system here. So even studying simple molecules can be extremely difficult using classical computing technology. Now, if we go to something more complex like benzene, the electronic correlations are so complex that, in fact, these kind of systems have been proposed to be good candidates for studies using quantum computers. Goings and others have said that understanding complex chemical systems such as biomolecules, catalysts, and novel materials is a central goal of quantum simulations. And quantum computing's potential to provide solutions to complex electronic structure problems has drawn considerable attention. Now, I think it's incredibly important from a fundamental point of view to study biological systems because they span what is known as the quantum classical divide. It's still an open question in quantum theory in terms of what really distinguishes quantum systems from classical ones. There is traditionally, in the orthodox view, a formal distinction between a quantum system of interest and the classical apparatus with which it is measured or observed. But of course, all matter is made of atoms, electrons, protons, neutrons, and other fundamental particles. Now, usually we can safely say that the classical physics applies to any objects about 1 microgram or heavier, and that quantum physics applies for objects in the zeptogram range. Now, this leaves a 15 order of magnitude gap between the quantum world and the classical world, according to Bassi and others in this review of modern physics I've cited. And so that really leaves open questions as how to how to negotiate the quantum classical divide. I've shown here in this figure really what that divide spans in terms of biological systems, all the way from simple biological molecules and increasingly complex ones like genes through proteins, and then through organelles, cells, and finally tissues. This is really what we're looking at in terms of the gap between systems which are clearly quantum mechanical and systems which are clearly classical, at least at the time of Bassi and others writing in 2013. So where is the quantum in quantum physics? Well, typically this is expressed in terms of Planck's constant, h, the so-called quantum of action that relates the energy of a photon, for example, to its frequency. Now, this quantum effect of the photoelectric effect was first discovered by Heinrich Hertz, and this is something that led Einstein to propose the quantization of light. Hertz had an electromagnetic oscillator, and he was able to use an induction coil to produce a primary spark in that oscillator. Now, what was beautiful about the experiment is that he was able to show that the first spark could initiate a second spark in a distant metallic circuit. Now, this is exciting because not just any color of light could produce the second spark. The first spark in the ultraviolet regime was necessary to overcome what's known as the work function in the metal in order to produce a second bright spark. Now, I have to admit that the creation of sparks has come a long way since Hertz's time, if you've ever seen one of the fascinating shows by Dr. Medicvault. And today, in fact, we know that quantum electrodynamics is the theory of the interaction between light and matter, the strange theory of light and matter, as Feynman called it, and it describes all forms of light emission and absorption. Now, these kind of effects are common in biological systems, like our jellyfish shown here, where we find fluorescent and phosphorescent effects. Electron transfer that is modeled using Fermi's golden rule, which of course derives from quantum electrodynamics, is very important in biological systems, as well as the theory of proton and electron tunneling. The radical pair mechanism is a favorite of quantum biological studies, which is derived from spin chemistry, as well as theories of super and subradiance, which have recently garnered attention in my own work and in other studies. As well, infrared and terahertz molecular interactions are increasingly gaining attention for their significance in biological processes, possibly such things as protein recognition. Now, the theory of ultra-weak photon emissions, these are biophoton emissions, becomes important because these kind of photon emissions are intrinsic to metabolic processes in all known forms of light. These comprise faint emissions in the UV through visible and infrared range. They're emitted by all living cells, and they're quite significant because they're a result of metabolic processes and cannot simply be attributed to heat or the kind of typical infrared emissions that we see from objects at ambient temperatures. Now, these ultra-weak photon emissions are implicated in oxidative stress, reactive oxygen species production. They're implicated in UV light resilience, which I'll talk a bit more in a moment. The mitogenetic effect, the purported ability for light to stimulate growth in living cells. They've also been implicated in microtubule dynamics, and they may play a role even in cellular communication. Both Niels Finsen, the founder of light therapy, and Niels Bohr, one of the founders of quantum mechanics, believed light to be an essential aspect of many physiological processes in living cells and tissues, and we now know that this is in fact correct. Photochemistry is incredibly important in proteins. Proteins contain many aromatic molecules, such as tryptophan, shown here, and these aromatic organic amino acids often have important photochemical properties and electron transfer properties, which are essential to the processes of light itself. Coherent exciton transfer and superradiant emissions have been implicated in microtubule systems and also even in such things as amyloid fibrils. If you look here to the right on the bottom, I've shown the fascinating pi-stacked array of tryptophans that can be found in an amyloid fibril that you may know as a key element of the formation of amyloid plaques that are implicated in Alzheimer's disease. So these could also have significant medical implications. So photochemistry without light is really the basis for these ultra-weak photon emissions. Why do I say without light? Well, first of all, if we want to detect ultra-weak photon emissions, we need to be in a very dark place because these emissions are incredibly faint. Living organisms are basically photochemical batteries that they, if you're a plant, absorb light from the sun, and other animals, of course, eat food in order to gain energy. And the resulting metabolic processes, as I've mentioned, produce faint light. Now, ultra-weak photon emissions can also be stimulated by shining UV light on skin, for example. And it was found, if we look at this work by Zapata and co-workers, that although tissue does have a base level of ultra-weak photon emissions that are being produced all the time, if you shine UVA or UVB light on the skin, that this will produce photochemical effect and light emissions for perhaps as much as 20 minutes or maybe even up to an hour afterwards. This is due to the presence of photosensitizer molecules in living systems. These include porphyrins, melanins, and other photoactive molecules, which once they absorb a photon become excited and can go on to produce characteristic oxidation reactions in other biomolecules, phospholipids, collagen, elastin, other proteins, DNA, etc. And as these excitations ultimately produce subsequent oxidative reactions, the excited molecular species that result in turn produce photons, which contribute to the total UPE. And we see a fairly dramatic increase in UPE intensity as shown in the plot. Now, photosynthesis and photosensitizers go hand in hand. Chlorophyll and heme have been called two of the most important molecules on the planet. Chlorophyll, of course, absorbs light during photosynthesis, and heme is the well-known oxygen carrier in the blood, found in hemoglobin, for example. If you look at these two molecules side by side, you see they could look almost identical, the main difference being the different iron center found in each. Whereas chlorophyll has a magnesium atom, heme has an iron atom, a transition metal, in its center. If we look at the catalytic cycle in luciferase, well-known enzyme that is used to produce light in bioluminescent species, the firefly luciferin molecule docks with the luciferase enzyme in the presence of oxygen. This resulting oxyluciferin produces light and other waste products, and then the oxyluciferin is released and the catalytic cycle can begin anew. Now, this kind of process is familiar to anyone who has seen a firefly at night or one of the numerous bioluminescent species that can be found under the sea. Jellies, anglerfish, bobtail squids, copepods, all of these species use, well, in some ways similar, but in other ways very different forms of luciferin that is also found in various forms in bacteria, firefly, of course, plankton, mushrooms, glowworms, toadfish, and yes, even snails. So bioluminescence is found, well, essentially universally throughout the biological kingdom, both in very, vibrant, explicit ways in bioluminescent organisms, but as I mentioned, the basic process that combines a photoreactive molecule, typically some oxygen species in an enzymatic reaction, is common to all forms of life. So this really highlights the importance of quantum effects in coenzymes. Now, coenzymes are complex organic molecules that are important in most, if not all, enzymatic reactions, and the electronic structure of these systems is highly non-trivial. I've shown here coenzyme tryptophan tryptophyllquinone, which is an important electron transfer coenzyme. You can see in the picture here the TTQ cofactor is transferring an electron to a copper atom in a nearby blue copper protein. This is some work that I did with Delalande and coworkers in 2010. And it's just one example. Electron transfer between proteins is just one example of the many quantum effects that enzymes mediate in biological systems. If we look, for example, at a light oxygen voltage sensing protein, like the one shown here, it has an FMN cofactor. The blue light activation triggers structural rearrangements that enable sensation. And although we know where the flavin molecule here sits, and we basically know what it does, the specific details of electron transfer in the binding pocket where the flavin sits are still poorly understood. Now, flavin mononucleotide is an important cofactor involved in responses to blue light. Flavins contain isoaloxazine rings, And in this case, the singlet and triplet dynamics have been identified in these kind of systems using, for example, time-dependent DFT, that's density functional theory, and models of spin-orbit coupling. Now, flavin mononucleotide is just one example of flavin types of molecules, such as riboflavin, which we all know from vitamin B2, and flavin mononucleotide you see here again, as well as flavin adenine dinucleotide, another favorite molecule to study in quantum biology. Now, all of these flavins have what's called an isoalloxazine ring. It's the functional group in all flavin enzymes, be it photolyses, phototropins, cryptochromes, amine oxidases, dehydrogenases, monooxygenases, and of course the blue light-using flavin proteins. Now, I'm sometimes asked why we can't just use quantum mechanics to predict certain physical and chemical properties of biomolecules, and then from there simply use classical models. We can effectively parameterize our classical models using simulations, ab initio type modeling, and then put our quantum mechanics textbooks away, so to speak. Well, I couldn't say it any better than Bayardi and coworkers have. They say, unfortunately, the underlying model potentials require sophisticated parameterizations and generally omit quantum mechanical effects, of course, once you've parameterized your classical model. They lack transferability to other model classes and therefore cannot describe well arbitrary chemical reactions and molecular interactions, if at all. We see here another common biological cofactor, nicotinamide adenine dinucleotide. And the problem with these systems is that we can use quantum mechanics to model the properties of it, but they really only give us the properties in very specific circumstances. If you change the molecule or just a few atoms of the molecule, or you change the environment, the setting in which it sits, you basically need to do your quantum mechanical simulations all over again. So simply doing one set of quantum mechanical calculations to parameterize a large number of classical models is rarely, if ever, feasible. So if we want to go from something like flavin, this isoalloxazine tricyclic ring, to physiological processes in living systems, factors such as the electrostatic preorganization, the charge distribution, protein dynamics, and active site solvation can all influence the reactivity of a molecule like flavin. Flavin molecules must include enzyme surroundings, but environmentally dependent functional properties of flavin continue to elude quantitative prediction. So even though we know we must include the environment, it's very difficult to say exactly how. In fact, there's only a very limited consensus on which quantum mechanical methods are suitable to simulate flavin photophysics. And even though flavin is known to interact with molecular oxygen, for example, acting as an electron carrier, flavin interactions with oxygen are poorly understood and are not predicted well by structure. Now, this is a really difficult situation because even though structure is a poor predictor in our current models of flavin activity, we know that structural modifications to flavin have a strong influence on its reactivity. So the structure is important, but we simply don't yet understand properly how. These kind of structural modifications are found in as many as 10% of native flavoproteins, so this is certainly something that is commonly exploited in biology for biological living systems to tune the properties of flavoproteins to the current need. Now, I haven't even really talked that much about metalloenzymes. So flavin, for example, is an organic cofactor, but these metalloorganic cofactors are also very common in enzymatic processes. And the electron structure of transition metal complexes especially are extremely intricate. If we look at the heme cofactor here, we have an iron molecule sitting in iron atom sitting in a porphyrin, surrounded by the molecules histidine to help coordinate its structure on either side. If we look at something significantly more complex, like the iron-sulfur cluster in nitrogenase, shown here, this carries out the agriculturally critical reaction for fertilizer production, for example, of nitrogen gas, the more or less inert nitrogen molecule found in the air, is converted to ammonia. Now, in this work by Lee and co-workers, two different levels of density functional theory methods, and more importantly, statistical fluctuations of the iron molybdenum cofactor reaction center at finite temperature have generated a well-supported mechanism over the entire catalytic cycle of nitrogenase. Now, if we look at that cycle, if we look at the FEMO cofactor, here it has a two-sided structure, if we look at the top right of the figure, and a less symmetrical one-sided structure where two of the hydrogen atoms have migrated from the top of the structure shown here to the bottom of the structure. Now, getting all these hydrogen atoms, these protons, centered in the right place is very important for actually producing the electrostatic forces in light of fluctuations to help pull the nitrogen molecule apart. So if you see the sort of dumbbell-shaped nitrogen molecule coming in on the left-hand side, if we go to the middle top, we can now see it can begin to form bonds with the FEMO cofactor, and centering itself, coming into the cofactor, now the forces become necessary to begin to break those bonds and pull the otherwise very strong triple bond bonding the two nitrogen atoms together apart. Now, this really speaks to the kind of sophisticated and concerted processes that are necessary for biological catalysis. If we look at another example, this is a pair of hemes in an enzyme that is responsible for making the TTQ molecule I showed on the previous slide. In fact, there's what's called a charge delocalization and resonance stabilization effect, where the electron is delocalized a long distance between the two heme cofactors, and this kind of delocalization is mediated by an aromatic molecule, in fact, a tryptophan residue in between. This is very important for carrying out the oxidative steps that are necessary to create the TTQ cofactor that is subsequently used in biological electron transfer and other processes. Now, this is very interesting because to create this long-range delocalization is effectively spatial entanglement is the kind of process that often is not expected to be found in biology, let alone to be found critical to many biological processes. But it's increasingly being shown that this kind of delocalization is essential not just for catalysis, but for energy transduction in living systems. So this sort of resonance is also important. It's now being determined in electron transfer flavoproteins, so-called ETFs, where it's believed that quantum non-locality is exploited to enable electron bifurcation during respiration. Now, if you've studied cellular respiration, electron bifurcation was a process originally proposed in Peter Mitchell's Q cycle, by which two electrons can enter the system with the same energy, sharing energy between them equally, and one of the electrons is bumped up to a higher energy level. When conserving energy, the other electron loses energy going to a lower energy level. This kind of energy splitting effect is commonly seen in spectroscopy, and in fact, bifurcation is crucial for survival in all known forms of life. Now, this is a fairly difficult kind of form of quantum effect to study, and it transcends the usual simplified models that are most commonly used in density functional theory, where we need to go to a more complex configuration interaction effect, where we actually model the interaction between the two configurations, the electron localized at one FAD in this picture or the other FAD, in order to fully account for the charge delocalization effect, which is now believed to play an important role in the bifurcation process, which now allows us to take two low energy electrons and get one high energy electron out of it, which can then drive subsequent biological processes, metabolic processes. Now, what happens during this kind of non-local charge resonance stabilization? If we look at a system like the photosynthetic special pair, in this picture, the electronic coupling is assumed to be the dominant factor, so that the charge becomes equally spread over both chromophores. And so if you imagine it in terms of an electron transfer where the reactant free energy surface describes the electron at the donor, one member of the pair of hemes, and the product surface describes the electron at the acceptor, the other heme in the special pair. The coupling is large enough that the electron doesn't simply tunnel back and forth between the two hemes, or chlorophyll in this case, pardon me, that in fact the coupling is now large enough so that the ground state of the system is delocalized across both molecules. So if we now consider not electron transfer resonance, but an effect where there's weaker coupling between the donor and acceptor molecules, then we can get more traditional stochastic tunneling effect, where in this case, for example, the flavin absorbs a photon, and the excitation now allows an electron to be transferred from one tryptophan, and in fact, it produces a whole cascade of electron transfers from a chain of tryptophan residues. Now, what's interesting in this system is that even though we're not looking at non-local resonance phenomena per se, that the whole process of these electron tunneling reactions actually involves a series of structural adaptations which are both directed and timed. So the whole protein, the whole photolyase enzyme actually moves in response to this kind of effect. And this gives some idea of how important the environment is in this case, as the now transferred electron is fired, so to speak, into a DNA lesion, breaking up the DNA lesion, and then the electron must be recovered subsequently to avoid causing further oxidative damage. Now, this kind of system with biological electron tunneling is also found in hemes, in system shown here, where a physiological electron transfer in the enzyme cytochrome B03 is important for again, energy transduction in the living organism. Now, in this system, instead of creating a resonant effect between the two, we have weaker coupling, still mediated by an organic residue, that produces a stochastic tunneling effect. Now, this, if you are familiar with Marcus theory, is reflected in the familiar pair of parabolas that are shown in practically every text on Marcus theory of electron transfer. Now, in this case, the electron can tunnel from one potential weld, the reactant free energy surface, to the product free energy surface. And because the electronic coupling is relatively weak, in this case, we don't see a sustained resonance. We don't see sustained delocalization between the two surfaces. Because the energetic degeneracy that's goes part and parcel with energy conservation only occurs near the crossing point of the two surfaces, and we don't see a shared lower energy state where the electron is delocalized between both the reactant surface and the product surface. Now, this tells us something fundamental in quantum mechanics. It's often asked, why don't we see macroscopic superpositions? Well, in fact, at the macro scale, every state we look at, it's not a microscopic quantum state. It's not a microstate according to statistical mechanics. It's a so-called canonical macrostate, an ensemble of rapidly fluctuating states that are known as the equilibrium state of the ensemble collectively. Now, if the free energy minimum, the so-called equilibrium state in our reaction coordinate picture, involves strong enough coupling between the donor and the acceptor, we can have sustained delocalization between the two surfaces. But if the coupling is relatively weak, then the system samples, for the most part, along one surface, the reactant surface, near the free energy minimum, before occasionally, very sparingly, reaching a point far enough along the surface that it can now enter very fleetingly a delocalized state before relaxing again to the product surface. And so this really shows, without invoking any kind of classical apparatus or even a measurement hypothesis, measurement postulate of quantum theory, shows why in one system we can see sustained delocalization, and in the other system it produces more of a apparent stochastic hopping effect, the so-called quantum jumps that Erwin Schrödinger so despised. Now, if we look at an even more complex system, like complex 1 of the mitochondrial electron transport chain, in this case we see a chain of iron-sulfur centers that regulate electron tunneling rate, synchronizing it with conformation-mediated proton pumping, enabling efficient energy conversion. And the synchronization of these rates is a principal means of enhancing the specificity of enzymatic reactions. Similar to the slide I showed on DNA photolyase, in this case, synchronization of changes in the protein conformation are essential to its function. So I've shown here this overall protein structure from this Science article. And if we take the protein ribbon away and zoom in a little bit, we can see, in fact, 8 iron-sulfur clusters, as well as a familiar isoalloxazine ring, among other organic residues. So it's very fascinating because in order to control electron transfer rates from nanoseconds to milliseconds, a chain of 3 or 4, or in this case 8, iron-sulfur centers might suffice. For example, these are found in enzymes such as succinate dehydrogenase, fumarate reductase, formate dehydrogenase, nitrate reductase, hydrogenases, and nitrogenases. So they're in all kinds of enzymes. In these enzymes, electron transfer is coupled to protonation, which must be properly matched to prevent formation of energetically unfavorable intermediates, slowing down catalysis, or avoid production of highly reactive intermediates. Proper timing is achieved by redox tuning, similar to the kind of tuning I showed on the previous slides, where the amount of coupling is tuned in order to produce the correct level of delocalization. In this case, however, the tuning is necessary to prevent the formation of toxic singlet oxygen species by the long electron transfer chains. And now further studies of this system have shown that the unique role of iron-sulfur clusters as electron transfer cofactors is in merging protein-water fluctuations with quantum state multiplicity to allow low activation barriers and robust operation. And so there's a number of fundamental factors which need to be controlled. The redox energy levels of the cofactors, the quantum mechanical coupling between the cofactors, fluctuations due to the presence of the solvent environment, and ultimately the state multiplicity, which is so important for understanding free energies. All are important for robust operation in this kind of highly complex quantum system that sits in the presence of ambient noise. Now, this brings me to the topic of biomolecular simulation. Biomolecules in their environment are extraordinarily complex systems, and simulation methods extend in order of complexity from basic density functional theory or Hartree-Fock simulations to include van der Waals forces and many-body dispersion effects. Then we need to include hybrid quantum and molecular mechanics simulations, possibly quantum embedding theories, and even dynamic mean field theory, which I'll talk about only briefly. I mentioned configuration interaction. When you can't just simulate it with one simple, fairly local electronic configuration, you need to go to what's called a coupled cluster approach. And this is all without even going to far more complex non-Hermitian or Green's function methods in order to analyze systems that are open to their surroundings. So if we start with the Hartree-Fock approximation, this assumes that the antisymmetric multi-electron wave function can be represented as a single Slater determinant. Now, in this case, we might actually choose to use something like density functional theory in order to provide an even more simplified, effectively trivial view of the quantum effects, where we model the system of electrons, the electron density, so to speak, as if it were a single electron or an ensemble of non-interacting electrons. Now, this gives us a potential in which we can treat the energy of the system in terms of an external potential, an effective Coulomb potential between the electrons, or which accounts indirectly for the interaction between the electrons, which are now treated as if they were not interacting. And finally, a so-called exchange correlation potential, shown here as a VXC, which is effective potential, an effective classical potential, which is aimed to account for all otherwise quantum mechanical effects. Now, even though it looks like in principle we can now simulate the consequences of a highly complex quantum mechanical interacting system with a single quantum mechanical system interacting with a series of effectively classical potentials, unfortunately at this time there's no formal solution to find the so-called exchange correlation functional. Now, quantum computer scientists, in fact, have proposed that finding the classical effective potential is, in general, just as hard as solving the hardest problems in quantum computing, a complexity class known as quantum Merlin-Arthur. So it doesn't seem like we're actually going to find an easy answer to the otherwise seemingly trivial problem of finding classical potential that reproduces all otherwise quantum effects. Now, as a result, there has been a proposal for a ladder of exchange correlation functions that can be developed to provide increasingly accurate approximations to the true quantum mechanical effect, sometimes known as quantum chemical heaven, where the ladder is shown as a kind of ladder or stairway to heaven of perfect quantum mechanical accuracy. At the bottom of the ladder, we have Hartree world, where the Hartree-Fock approximation is the only approximation that's really used. This can then be extended to what's called a local density approximation, where correlations are given by the local electron density. A generalized gradient approximation, where now we assume that, in fact, the electron correlations can be predicted using the local density as well as the gradient of the density. Now, the so-called meta-GG methods expand on the generalized gradient approximation to include the kinetic energy of the electrons. And from there, increasingly advanced hybrid approximations must be used that combine the true exchange correlations with various DFT approximations. From there, we go to double hybrids, and we might include something like a Muller-Pleasant theory in order to account for various corrections, the so-called random phase approximation, and still have a long way to go before we get to so-called chemical heaven. Now, if we then imagine following this stairway from the simplest approximations to the most challenging ones, the electron delocalization is first treated with van der Waals forces and dispersion. Now, these kind of true quantum mechanical effects sometimes can be treated with relatively simple parametrized models. They're very important for things like the pi stacking of aromatic molecules, and of course, they're significant in any solvent system. Now, in DNA in particular, the electrostatic repulsion between stacked base pairs is balanced by the attractive London forces, these van der Waals forces, and these are essential in order to, in your model, to get the structure of the DNA double helix correct. These London dispersion forces are also believed to help enable wave-like electron transfer through DNA, as well as its structural stability. And these make DNA a very interesting quantum material because it has good electron conductivity, but comparably low thermal conductivity. And this is unusual factor to have good electrical conductivity, but rather poor thermal conductivity. But it's certainly something that gives the DNA its very interesting quantum mechanical properties. Now, if we want to go beyond these kind of picture to a multi-scale model, there's a number of techniques that have been developed, and Nobel Prize that was given out for multi-scale biomolecular models, where we have a quantum mechanical model embedded in a larger, effectively classical model, and perhaps even a classical continuum model. Now, despite claims to the contrary, biological systems often contain highly complex quantum effects at their core, and conventional quantum chemistry methods often fail to account for highly complex effects. And so this has driven research on the use of hybrid models, where the quantum mechanical part might be parameterized using machine learning, high performance classical computing, or even proposals to use quantum computing to solve the hardest aspects of these biomolecular problems. Now, if we look at these mixed quantum classical models, the so-called QMMM models, these combine classical force fields with quantum simulations, and these can be used to study dissociation constants and redox calculations, ground state structure and biochemical reactivity calculations, as well as studies of spectroscopic and other excited state properties that are reflected in the structure and dynamics of these systems. QMMM methods then can be extended to cover mixed quantum classical dynamics. And now these are important to study ultra-fast non-adiabatic processes, such as internal conversion and intersystem crossing, where the so-called configuration interaction effects become essential to the process, and other forms of non-adiabatic state transfer and surface hopping processes, similar to the electron tunneling processes that I discussed on some previous slides. If we look at how to model these complex quantum effects, for example, in the hemoglobin metalloprotein, it's essential, for example, in hemoglobin to study these properties in order to adequately characterize its ability to store oxygen and to properly distinguish its interactions with something like oxygen to another molecule like carbon monoxide. Typical quantum chemical methods like density functional theory are so far unable to provide a complete description of binding of molecules like oxygen to heme, and problems with simple quantum approximations can be addressed by accounting for, again, increasingly complex effects, both strong local electronic correlations. This can be addressed using dynamic mean field theory, quantum non-local entanglement, which is addressed using coupled cluster methods, and other advanced multi-reference electronic methods. So if we want to study both the dynamic and the static correlations, we would then combine density functional theory with the so-called Hubbard on-site U correction, and then we might extend that further to include aspects of dynamical mean field theory, which in a mean field theory approach include dynamical effects that are intrinsic to, for example, van der Waals forces. In order to address this kind of system, we would use a frequency integrated Green's function, like the one shown here from this paper by Weber and colleagues. And if we need to further deepen our understanding of the system, we might go to a multi-reference method like the complete active space self-consistent field theory, which in turn can be combined with DFT and DMFT, and might even be further parameterized using calculations using density matrix renormalization group. So the list of these increasingly advanced quantum mechanical methods goes on and on and on. And that's before we get even to the photobiophysical modeling of these systems, because of course many biological processes, as I said from the start, absorb and emit light. Now in this case we need to properly account for the system's interaction with the so-called quantum vacuum. Light can influence photochemical resonances, structural rearrangements, and even increasingly refining molecular forces. And this requires, again, a so-called Green's function approach in order to properly account for the system's interaction with its environment. And it can be incredibly important if the system is strongly interaction. This is the kind of thing we typically see in cavity quantum electrodynamics, but can be reproduced in principle in a biological system through the presence of thin films or a high density of structurally organized chromophores, which of course are universal in biological systems. So understanding quantum mechanical resonance involves not just this kind of open quantum systems approach, but a deep understanding of how losses from the system and noise or energetic driving effects can counterintuitively play a synergistic role in these apparent non-linear, non-Hermitian resonators that we see all over the place in biology, in microtubules, in photosynthetic complexes, in the pi-stacked arrangements we find even in amyloid systems and in a variety of photoreceptor systems that all are found in practically every form of living organism in the world. I've given a few references here, both from my own work and other fundamental research papers studying non-Hermitian effects in this system. And I direct you to the Hamiltonian I show here. This is a classic PT symmetric kind of Hamiltonian that can produce singularity, where we might see dramatic response from the system to comparatively weak stimulus, which of course has been this kind of investigation into large responses to weak stimulus has been a classic problem to be investigated in quantum biology. So I'd like to wrap things up with a few challenges for quantum biology. I believe that quantum biological systems pose key challenges for our understanding of the environmental role of photophysical interactions in biological systems, for our understanding of electron transfer and transport synchronization in biological systems. We also see challenges in our understanding of catalytic enhancements for biochemical synthesis. We've talked about regulatory tuning of coherence and decoherence in these resonance stabilization and tunneling effects. Biological systems seem to be able to perform direct manipulations of spin selective phenomena and photochemical modulation of metabolic processes are now well documented. Finally, these biological systems seem to be able to exhibit fairly exacting control of spontaneous symmetry breaking effects. Again, I talked about the transformation from the charge resonance stabilized state, where the electron is equally delocalized across a pair of electronic cofactors, to the symmetry broken state, where the electron spontaneously hops back and forth between the two cofactors. So all of these kind of speak to both challenging questions in quantum simulation, but also some fundamental questions that speak to the heart of our understanding of quantum mechanics itself. So in conclusion, I'd like to say that we can approach the study of biological systems from first principles, and that this presents new possibilities to advance quantum physics theory and to inspire new biophysical applications. So in closing, I'd like to thank you for your attention to this talk. Of course, I'm always happy to take any questions to my email. Please continue to check out my YouTube channel at Dr. Babcock. And finally, don't forget to check out my new monograph, Physical Principles of Quantum Biology, which is on the Physics Archive and can be found simply by snapping a picture of this QR code here. Thanks again, and have a great day.